In this video, I am talking about chemiosmotic theory. This will be help us to know that how exactly ATP will be synthesized in cyclic and non-cyclic pathways. Let us draw one thylakoid. See what will the present here so that we can understand the exact process of ATP synthesis. Now we know the exact structure of the thylakoid. Let us draw the photosystem. The photosystem, that is PS1, PS1 and PS2. The electron is going to pass from PS1 to PS2. That is pass from PS1 PS2, sorry, this is PS2 and this is PS1. The electron will be going from PS2 to PS1. So say this is a cytochrome. So this is a cytochrome. When sunlight is falling on the PS2, all pigments, here is many pigments is present, all pigments absorb photon. What absorb is proton, and electron will be excited. Here is present electron, this will be excited and it will be taken to the primary electron transporter and it pass to the PS1. Then it will be release energy, and when the electron is passing through the cytochrome, this is cytochrome, when the electron is passing through the cytochrome, at the time except a pair of proton which is present here, which is plus from the stroma, this is the stroma and using that energy it pumps that pair of protons to the from the stroma to the lumen. So it pumps the protons from the stroma to the lumen. Actually, what does the cytochrome do? It will accept the proton and transfer the electron. It will transfer the electron. So it is a proton acceptor and electron transporter. This is part 1. Secondly, to replace the electron pair of the photolysis of water take place. Here is photolysis of water take place. So now this water split here, and so splitting of water take place in the lumen. This is the lumen. So three things are released, that is a pair of electron and which is given to PS2, a pair of hydrogen and half of oxygen, one oxygen atom. This is the byproduct. Oxygen is the byproduct. So O2 is produced in the lumen and then it will be diffused out and given as byproduct. So now the proton which is produced due to the photolysis of in the lumen and the proton which is pumped into the lumen from the stroma, they are also accumulating in the lumen. So here is 2 proton is found and here is from the stroma 2 proton is accumulating. So there is a accumulation of proton in the lumen and the reduction of proton in the stroma. So there is a true process. Photolysis of water is taking place and the lumen is highly concentrated with proton. This proton will tend to come back in the stroma because here is accumulation of the proton, so it will be transported to the stroma and they can come back only through certain particles. So I'm going to tell you that what type of particle is here. So here is some type of protein is present. Here is a protein present, and this is known as F0 particle, and this is the integral protein embedded in the plasma membrane. And there is another type of protein protein which is extrinsic protein. This is intrinsic protein, this is extrinsic protein, which is be protruding out from the membrane, and it is known as F1 particle. So this is a F0 F1 particle. Both are called as ATPs. The proton, they can come back into the stroma through this channel. The proton, which is accumulation of proton which is taking place here, this is come back only through the F0 and F1 particle. So F0 F1 particle act as a channel protein for the proton to move from the lumen to the stroma. As the proton is moved for the higher concentration to the lower concentration, they would also release energy. They will also release energy. F0 particle help in the facilitated diffusions of the proton. So here is no energy is required, and now when this energy is used by the F1 particle to add inorganic phosphate, here is inorganic phosphate is add and to ADP to synthesize ATP. So inorganic phosphate and ADP is bind with F1 particle, and this gives us ATP, and then it come back to the stroma. So this is the second part, and up next is I'm going to tell you the electron pair which is coming here will go to the PS1, then through the electron transport chain. So this pair of electron this pair of electron will go to the NADP plus, and this NADP reductase enzyme is going to help in the formation of reducing power. But for that, proton is required. From where the proton is made available? So proton which is coming here will joining here, and this proton and NADP 2H plus NADP with the help of NADP reductase enzyme gives us NADPH2, and this is the entire process of chemiosmotic theory. And hope this is helpful to you, and thank you for watching my video. Please like, share and subscribe my channel. Thank you for watching.